this course just happens to be the coolest course that you will take as an undergraduate. In truth, when I was an undergraduate at the University of Michigan, anybody want to guess what was the coolest course I took? Anybody?
Biophysics?
Yeah, it wasn't.
Chemistry?
Chemistry? No, it wasn't. It was constitutional law. It was really great. But I'm going to make biophysics be the greatest course you can take, because you'll learn incredibly cool things. Okay? In fact, if you decide to really learn these things, then eventually you can apply them and make, I don't know, better drugs for people whose bodies have gone south a little bit. Okay, today we're going to go through the basics of life. What elements make you up? Why those particular elements? What all life has in common? How you came from billions of years year old creatures. We're going to talk about the one great rule of molecular biology. So I'm just curious, how many people can tell me what the great rule, hint, hint, is? Raise your hand. So you would feel comfortable. Keep your hand up. There we go. Come on, you're excited. Yeah, no. I just want to see your hand. There we go. There we go. Before he was like very into it. You're a graduate student, right?
Yes.
Yeah. So okay. Okay, you can put your hand up. So anyone who raised their hand would be willing to tell me about what this rule is, and for example go on for a couple of minutes about what it is. Raise your hand if you're willing to do that. Oh, come on. Not that many. Okay. By next class, I want you to do that. Okay. So the great rule of biology that all of us follow is DNA going to RNA going to protein. And it turns out, because of this, it leads to the great theory of biology, and that is evolution. Often physicists come in here and want to know what the great theories of biology are, right? I mean, because in physics, what are some of the great theories that we have? Who knows? But by the way, I want you to participate. And if you're not going to participate, I'm going to be very sad. And more to the point, because I can, I'll then call on you. Okay, so what are some of the great theories? Yeah?
Relativity.
Relativity, okay. What else? Yeah?
Gravitation.
Gravitation, okay.
Wave-particle duality.
Okay, wave-particle duality. Boy, these people are serious.
String theory.
What about more ordinary things? Yes? Newton's laws, for example. How things move.
Energy conservation.
Energy conservation.
Thermodynamics.
Thermodynamics. Okay, what about in E and M? You know, there are four Maxwell's equations. I mean, in mechanics, there's 3 laws, and E and M, there's four equations that govern it all. In general, in biology, you don't have this. The reason is biology is largely a series of tricks. They started with some simple things. They randomly tried some things worked, some things didn't. When the thing worked, there was additional pressure on them, and then they did another series of mutations. And in that sense, for 4.5 billion years, things have evolved. And you'll see that actually they've all evolved from some sort of simple organism. Okay. Lastly, what we're going to find is something called the Boltzmann constant and what it tells about DNA stability. Who can tell me about Boltzmann's law? Who was Boltzmann? Yeah.
So Boltzmann gave equation for knowing the entropy of the system.
Yeah.
Depending on the various states it can take system.
Okay.
Energy states.
Yeah, that's good enough for now. And then we'll actually be able to do a little detail about this. Okay, by the way, it's 10 minutes after the start of class. Has everybody who plans on turned it in? Okay. Last one. Oh, got another. You always have a straggler. Thank you. That's it. Going once. Going twice. Come on, going once. Going. Hey, you got to help me out a little bit. So I'll say going once, and then you say going twice, going 3 times, sold. Okay? Going once. Going twice. Going 3 times. Sold. There we go. Okay. First, let's introduce ourselves and discuss some practicalities. Okay, who am I? I've already told you. I'm Paul. After office hours after class, 11 to 12 on Tuesday. Also, often it'll be after class on Thursday as well. There's my email and there's my telephone number. Okay, who are your graduate student TAs? Okay, we have Marco, Josh, and Chawi. What we're going to do is I've given them a minute each to tell you about themselves, and they'll maybe tell you about something having to do with physics, and then something just that's interesting. Okay, Josh?
Sure, I can start us off. So my name is Joshua Moss. I'm a 6th-year graduate student in the lab at Montecursion. I'm almost done. And what I like to do outside of studying membrane proteins and how they interact with the lipids around them and how they undergo conformational changes to transport them from one side of the membrane to the other, I like field hockey. And yeah, that's kind of the big story. If you have any questions, I'm here. My office hours, I'm sure there'll be for those, are on Friday.
Okay.
Joey?
I'm Charlie, I'm from China. My research interest is in neuroscience, in like getting the super resolution image of the neuron and how they talk to each other. And besides physics, this research, my interesting thing is like watching some sports games and yeah, I think that's it. And I hope you guys have a great semester with Professor Boltzmann.
And Marco?
I'm Marco. I'm a 7th-year graduate student with Paul now, hopefully finishing soon. My research interest, you are going to learn quite a bit from what Paul is teaching, molecular models, different things that work inside the cell. Couple of interesting things about me. I have a twin brother.
Okay, let's welcome. Okay, so they're welcome to stay for the talk. They're also welcome to go. Okay, there you have the office hours. The fixed one is from Friday 4 to 5:30. The other one will be floating on either Monday or Wednesday, depending on whether the homework is due Tuesday or Thursday. In general, I'll give out homework pretty much every week, except, there'll be weeks when we have vacations or midterms or finals and so forth. In general, the homework is due 1 week from the date of the assignment. So I will, on Thursday, assign you something. It will be due on the following Thursday in class, at the beginning of class. Okay. Okay, what about grading? Roughly, I'll give out about 45% A's, 45% B's, 5 to 10% C's. So in general, you should be able to do very well. In fact, if you really learn this stuff and you can show it to me, I'll give you an A+. I'll give every single one of you an A+. So you're not in competition with the person next to you. In fact, I strongly encourage you to work together. You will do much better on the homeworks and therefore on the tests if you work together. How will I tell if you've learned? The midterm will be worth 30%. The final will be worth 30%. The homeworks, as I say, pretty much every week, so there'll be about 10 of them, will be worth 30%. By the way, with the homeworks, what will happen is you are allowed to be able to miss one of them. So you have to do 9 out of 10, although the 10th one you're not allowed to miss. Okay? There'll occasionally be quizzes. In fact, 1 week from today, there will be a quiz. So you should be aware of this. And I'll tell you about that. Okay, there'll be about 5 of them, something like every other week. Also, I'll ask you to tell me a few questions about what happened during the past lecture. And I'll give you a few minutes. Okay. This was about the quiz. Okay. In general, you'll also be able to give feedback and evaluation of my lectures. At the end of the class, I will have you fill out a quick sheet as to, I'll tell you about it. In general, every lecture at the end, I will ask you for that feedback. What I will do is I will choose a particular person 6 times, and if you get 5 of them 6 for 100%, you'll get, this 5%. Okay. Yes, you get to evaluate the class. This is what you'll see at the end of the class. What was the most interesting thing you learned in class today? What are you confused about? Relating to today's subject, what would you like to know more about? And for any helpful comments. In fact, today, for the 4th one, I'll ask you, what subjects are you particularly interested in? Would you like to see? Because I'm willing to change the course somewhat for you. Okay? You answer this, I'll give you 5 minutes, and you turn it in at the end of your class. And then again, I typically start the class, the next class, with your previous questions. Okay. I said occasionally I'm going to give you a quiz. This will be your quiz number 1. I mean it. Literally, this will be your quiz. You will see this next Tuesday in class, and I'll give you about 10 or maximum of 15 minutes where you have to answer these. In general, I won't give this to you, but the first time I will. By the way, I see somebody is taking a picture. That's fine. That's just no. It's fine, but you don't need to take it, because at the end of class today, all of this will be on the web. Okay? Of course, if you want to take a picture, Anybody want to? Okay. Okay, who should be in here? Basically, the prerequisite is to get a semi-quantitative view of biology using the physical tools. As you mentioned already, there's a diverse crowd, actually, I haven't asked, how many are physics majors or something like that? Quite a bit. How many are biology majors? Not that many, but a couple. Okay. That's probably good. How many chemistry or something like that? Okay. So how many, I'm curious about this, how many figure that they know the biology, like the central dogma of molecular biology, DNA to RNA, so forth. Go ahead. Okay. How many don't? Come on. You can do it. Okay. That's fine, because I'm going to go over the biology. We're going to do it really relatively quickly, so then we can get to the physics behind it. In general, what I expect is you have at least physics 111 and 112, basic calculus, some knowledge of statistical mechanics, and in fact, I'll be going over the statistical mechanics somewhat. Okay. There's basically no assigned textbooks, though there is these on the web. Okay, what are you not going to learn? This here is the metabolic pathway of some snake doing something. I'm not going to teach you this. I mean, it's just unbelievable. It really is incredible. And it's great. If you want to learn it, you go talk to a biochemist. I'm not a biochemist. Okay? And I hate memorization. So forget about it. You will need to memorize certain basic facts, but I'll keep that at a minimum. Okay? Okay. So what are we going to learn today? So now we're going to start. Bye. Bye. I've lost 2 of them already. One is staying. Not so bad. Actually, before we get on to this, what questions haven't I answered? Any? Yes?
Are you going to treat grad students and undergrad students separately?
Yeah, that's a good question. Basically, graduate students and undergraduates will be treated the same. It's a little bit different in that, if a graduate student gets a C, that's essentially failing. So, try not to fail. Yeah?
Are all the lectures going to be recorded?
All of the lectures will be recorded. In fact, on the website, what you'll have is the slides. It's not me being recorded. However, see right here, they are recording it, and I don't know, does it get posted somewhere?
Maybe by the end of this coming year.
Okay, so you'll miss my wonderful voice. Okay?
I didn't hear his answer, sorry.
Yes, it will be recorded, but it won't be available until the end of the semester. So basically, no.
Okay, thank you.
Yes?
Is the midterm going to be in class?
The midterm will be in class. The final will be during the final time. So the midterm will be an hour and a half, and the final will be, whatever, 3 hours or something like that. Okay? Any other questions? Yes?
What will the homework be like? Will it be like what we just did with like answering problems from the text, or will it be like solving problems, or will it be both?
Yeah. Both.
Okay.
Yes. What are you eating?
Some cheese and bread.
I'm not that into that. So you can keep it. I don't have to. I mean, you weren't offering to share it? Oh, okay.
Yeah, so I'm like, okay, students eating during lecture, I'm like,
Go ahead.
Anyway, what I was asking is, for the syllabus that, I think this was a previous syllabus, but there was a distinction between written reports and another type of homework. Do we?
In fact, the previous syllabus is the previous one. There will not be any written reports. However, there will actually be 1 or 2 written reports that'll be part of the homework that I'll ask you to write about something. And it probably won't be about your dog. Who, by the way, I can't resist. Who has a dog? I have a dog that I generally bring in to school, and he's really great. Oh, she is really great. Next time I'll show her to you. Okay, I guess we should get a roll on. Okay, so what are you made of? Here it is by percentage of body mass. 4 elements make up 96% of you. Why? Water. Very good. Mostly you're big bags of water. Actually, it isn't just a bag of water. It tends to be salt water. So it has potassium, sodium, chlorine. Also, these are important ions for signaling between cells. Okay? Where does it? So, we've just gone and accounted for 93% of you. Yes?
Most of the air, like the atmosphere, is nitrogen, so we breathe it in.
That's a very good thought. What percentage of air is nitrogen?
16%.
Yeah, 60, 70%, or something like that. Huge amount of nitrogen. You don't use it.
It should be protein, I think.
What's that?
In protein amino acid.
Amino acids in proteins, yes.
DNA.
In DNA is also there, yeah.
I was going to say proteins.
Okay, proteins, DNA. That's basically correct. Amino acids, amino means nitrogen, also DNA. Where do we get the nitrogen from? It is not from the air. The air is N2, which is incredibly stable, and we cannot break it down. Interestingly enough, what can break N2 down? Or does the N2 just float around and never get into or biochemical system. Oh. Wait, yeah, this is a question.
Soil bacteria.
What?
Soil bacteria, nitrogen fixers.
The nitrogen fixers, that there's a class that can break down N2 into N and enter into the biochemical system. That's the only way. The N2 that you breathe in is completely irrelevant. Okay. Calcium. Turns out calcium is actually a metal. It's the most significant metal. Often it's as an ion, calcium 2+. The main thing is in your bones. In your bones are primarily calcium and what else?
Phosphorus.
And what?
Phosphorus.
And phosphorus. Very good. Fortunately, it's mostly in the form of PO4, which is phosphates. What if it was really phosphorus? Anybody know? What's that?
Burn.
Burn, yes. It's generally, it's not, generally it's called white phosphorus. If we had this, okay, so calcium phosphate. Also that it's used a lot in signaling. However, one of the things is osteoporosis. When you get low on the amount of calcium, what your body tends to do is it takes it from your bones and uses it other places. And then later, when it gets some more calcium, it then goes and puts it back into your bones. Often, there's a problem that it doesn't put it back into your bones, and that's called osteoporosis. And that's a big problem especially among older people. Okay, so one thing, we've talked about how your bags of salty water, but we haven't talked about carbon. Why is carbon such a big producer? Okay, what's your name? Katie. Okay, Katie. That's great, but I'm going to make sure that other people around you. Yes?
It's the only one that can, well, it's not the only one, but it's the most easier one to form four bonds.
Good. Excellent. And so what is it about 4 bonds that makes carbon so good? The thing about 4 bonds is it can form macromolecules. It can form a lot of like straight chain carbons, single bonds, or double bonds, or rings, or it can form all sorts of complicated molecules. Okay? And carbon is basically unique. Well, is it? What other atoms or elements have 4 valence electrons? Yes? Silicon. Good. I figured someone who would know this. So, So, why aren't we made of silicon? There's an awful lot of silicon. In fact, what is the most common form of silicon that's all around us? Sand or rock, silicon dioxide. Why aren't we, like, what is that? Hulk. That's right. So why are we all hulks? Well, naturally I am. Okay, now, so far we've had the same couple of people, and that's great. I really appreciate it, because being up here and talking, I I mean, I like talking, but it's nice to, occasionally. Oh. Okay, so what was I asking? Oh, silicon. Why aren't we made of silicon? So someone new. You've had your cheese and crackers, and so maybe I'm going to have a little of that. So save it a little bit. Okay, come on. So somebody who hasn't answered a question. Why don't we use silicon instead of carbon? Okay. La-di-da-di-da. Thank you very much. What is your name?
Lawrence.
Lawrence. My brother is named Lawrence, although more often we call him Larry. He's my older brother, so I sometimes call him an idiot. Oh, sorry. Okay, yes, Lawrence.
Just a guess, but it's like heavier, so it's not as useful.
No, that's exactly right. Silicon is a little heavier. So why is that right? So what about it being heavier means, why don't we have, silicon like we have carbon? What's that?
It can't interact. It will have lots of interaction.
Not really. Silicon is heavier, but because it's heavier, it's not soluble. You have multiple silicon dioxides, and therefore in water, it tends to fall out and be insoluble, as in rock. But in fact, if it's insoluble, why is things being insoluble not good? Because, oh, yes?
I was just going to say, because most of the reactions that takes place in our body happens in solution.
Yes, most of our reactions in our body takes place in solution. And why is that so important? Why is that? Not supposed to ask that. Why is that?
Because water plays an important role in these reactions.
Yeah.
So it can be moved around in the body?
So it can be moved around in the body? Oh. Well, we apparently don't know the answer. Because it needs to be in equilibrium with the water molecule. In fact, so it can transfer, it can do some reaction depending on whether the environment has changed or whatever. And life is all about, reacting to the environment. So it's pretty amazing. What we've done here is we've accounted for 96%, in fact 99% of the atoms in our body with just a little bit of thought. Okay, so they have, yes?
On the previous slide, there was a term called nucleon.
Yeah. The nucleus. How many protons and neutrons there are. So carbon is typically 12, and silicon is 28, and that sort of thing. Okay, these carbon and similar elements, like nitrogen and so forth, typically make up they make up small molecules, and then the small molecules come together, and they form 4 macromolecules. And the thing about life at this level is basically you're made of 4 macromolecules. One of the macromolecules is DNA. Also, it can be RNA, which is somewhat of a difference, which we'll get into. DNA is this famous twisted helix. 2 helices come together, and then they twist up. And in fact, they're made of a series of nucleotides. that looks something like this. And now we'll go through some details. The second is proteins. In fact, DNA makes RNA, which then codes for proteins, and these are made of amino acids strung together to make a protein. They're strung together in a linear form. There's amino acid 1, amino acid 2, and they're strung together, and amino acid 3, they're strung all in a linear arrangement. There are no what's that? Branches, that's the word I'm looking for. Branches. Okay? And it turns out that there's 21 different amino acids, and the difference between them is this so-called R group. And we'll talk about that in a little bit. Third, among the 4 macromolecules are lipids. And lipids is sort of a fancy term for generally fats. It has a lot of these carbon linkers, which in general are not soluble in water, and they're really good at creating membranes, which are little vesicles where you have things like proteins and DNA inside of them so they can do their own special reactions. And what's the fourth one? Who can tell me? Yes?
Oligosaccharide, like sugars.
Good. There we go. That oligosaccharides or polysaccharides, the word saccharides is just another word for carbohydrate. So that basically they're made of simple sugars. This one is glucose, and they're made, these are conjugated together, and they make big ones. Okay? Okay, all life follows the same basic rules. This is actually a big concept. What is that basic rule? DNA to RNA to protein. That's called the central dogma of molecular biology. Francis Crick, of the famous Watson and Crick pair, discovered of the DNA double helix, made it up. The thing is, actually he called it the central dogma of molecular biology, and it's that you have some DNA, which makes some RNA, which then makes some protein. He actually, in retrospect, hated the word dogma of molecular biology because he said, well, this is science, and so science, it has to be very rigorous, and so it doesn't matter how well known the facts are, it should be amenable to examination and change. And in fact, we'll look at some of the changes. For example, I asked you in the homework what siRNA is. Yeah? So how many people, I'm not going to call on you, but how many people answered it and feel like they know what siRNA is? Raise your hand. Yeah, I like that. A couple of people sort of look around and say, how many people? Yeah, a lot of them. Okay, I'll raise it. It's very clever. Okay, so DNA is the genetic information that is very stable. It lasts an entire lifetime. So something about it has to make it such that it is very stable, such that the information involved in this is stable over your lifetime. So you can produce kids that are like you. In fact, what it does is it takes advantage of some of the lipids in that it makes its own special compartment called the nucleus to protect it. And then it has to make an RNA, which is like a copy of itself, but it's somewhat more reactive than DNA. In fact, it makes it in the nucleus and then transports it out of the nucleus in the cytoplasm and where it can do its stuff. And in particular, the RNA can be used rapidly because it needs to react with other things if the outside environment changes. What it is it's good for, it copies the DNA into what are called proteins, which is basically the workhorse of the cell. It does everything. Basically makes structural components. It does catalysis, which is, in other words, in biology, generally we call enzymes, which make reactions go which normally don't. And we'll discuss the business of enzymes and how they work. Okay, if all life is based on the same fundamental rule or rules, what can we say about the relationship among all life forces, life forms? And what we can say is that all of life is related. That you and you and you are all related. And that, okay, we call you humans. Though humans are related to monkeys, because you rely on the same basic laws as monkeys do. Just like you look a lot like your parents, and therefore we figure you're related, you kind of look like the monkey. Though we can go farther than that. The monkey is related to whatever, the baboon. And the baboon looks kind of like a squirrel. I'll make some up. And the squirrel is kind of like a cauliflower. Actually, it's true. A squirrel goes from DNA to RNA to protein. So does the cauliflower, from DNA to RNA to protein. It turns out, so anybody know what genes are? What's your name?
Alex.
Alex, okay. Alex is one of the people who are great, that I really love, who he knows the answer and he raises his hand. What's actually best is actually the people who don't really know the answer, because then we have an interesting conversation. Okay, so what is the genes? You've answered before, haven't you? Yeah, sorry. Okay, there we go, yes. What's your name?
Michael.
Michael, okay. Wait. You know, we have to get to know each other. So, what's the deal?
Like a section of DNA that...
No.
Okay.
You
My arm?
yeah. Let's get something interesting. It's messed up. Okay. Michael has a messed up arm. Go ahead. You're telling me about DNA. No.
DNA that codes for proteins?
Yeah, okay. That's right. And you have this big, long DNA. About how many base pairs, how many of the little steps does DNA have? About 3 billion of them. In fact, and they tend to be divided up into about, for us, it's about 46 sections. Each section is called a chromosome. And within that chromosome, there are parts of the DNA which actually code for protein. There are other parts, actually it turns out about 90% of the DNA that you have don't code for proteins. In fact, it was a big mystery up to about 10 years ago what this other DNA did. In fact, it was called junk DNA. Turns out, actually, that's totally wrong. Actually, it's really important, and we'll we'll get that later. Anyways, so what was I saying? Genes. What was I saying about genes? I was talking about you versus the cauliflower, that you're related. Both are going DNA to RNA to proteins, and it turns out the number of genes that code for you and the number of genes that code for a cauliflower has about 50% similarity. That's pretty amazing. So when you're about to eat a cauliflower, you know, like, it's like you see the cauliflower, don't eat me, don't eat me. Okay, it's possible, though unlikely, that this cauliflower you're talking to independently arose this procedure to make DNA to RNA to protein, and so did you. It's possible, but it's pretty unlikely. In fact, so that says we're all related. At some point, the cauliflower had a certain DNA and RNA and protein. It eventually mutated, and boom, out you came. So think about it. How many of you are vegetarians? Not too many. So it seems like 3 out of 4 of you were Indian or East Asian.
Does it count that I used to eat meat?
Does it count that you meat?
You want to be vegetarian all?
I don't know. I'm just making you vegetarians who are generally somewhat above the rest of us who eat meat. Say like, oh, you see, I'm not killing anything, or just forget it. Okay, so you vegetarians are better than the rest of us. I partly believe that. I love eating meat, but I'm trying to become more like you vegetarians. Okay, anyways, God, okay, so does biology have any great theories of laws? And that is Charles Darwin. He made The Origin of Species, and he said that everything, all of life, is based on evolution. Take a simple organism, it undergoes some random mutation. It can go from a cauliflower to, I don't know, broccoli, goes to an eggplant, goes to a little bacteria, goes to maybe a multicellular thing, and eventually can walk and so forth. Okay? So it says evolution, that life basically evolved from simpler things. Evolution is no joke. It's one of the best tested scientific theories around. Okay, now we're actually going to talk about what the central dogma is in a little bit more detail. What you have is, you have the nucleus of a cell and you have the cytoplasm around that. Each are defined by lipid layers. In the nucleus is where you want the DNA to be. You protect it from a lot of the things that are going outside. As I mentioned, DNA is a double-stranded helix. It's made of 4 nucleotides, which we'll just say that T, which stands for thymine, A for adenine, C for cytosine. Well, here's another C. What G is for what? Guanine. Okay, so good, you know it. Okay, and they're arranged in some order. In general, they form a double-stranded, so the reason is they're less likely to be altered because you want the thing to last a long time. On the other hand, the information in this happens to be the order of the nucleotides. This is TACC, and another one may be TACT, so forth. Okay, in order to react, it has to make another biomolecule, which is basically identical to this, and that is it makes RNA. And it makes an A, and then a T, and then a G, and then G, because there are certain laws, namely T binds to A binds to T, C binds to G, and so forth. Okay, and this process is called transcription, and you make a single-stranded RNA. The RNA is made, and it moves out of this nucleus and into the cytoplasm. And once it's in the cytoplasm, over here, then it goes and interacts with another protein plus actually RNA, a complex called the ribosome. And here, the ribosome essentially reads the RNA. And it reads it every 3 base pairs. AUG, that's 1 unit of information. Then GCU, that's another unit of information. What is that unit of information? It turns out these 3 AUG code for 1 amino acid. And then GCU codes for another amino acid. And it turns out that there are about 21 different amino acids. And it's the ribosome's job to take this RNA sequence and to transfer it into amino acid sequence. And then you have this big, long amino acid, and that is called your proteins. Eventually, it gets to the end. The protein, which is, again, this long string of amino acids, folds up into a structure. And when it's folded, we call it a protein. OK? Is there any questions about the molecular biology of the cell? Okay, it turns out that there's actually a lot of questions associated with this. And in fact, we'll spend quite a bit of time on this. Okay, so let's spend just a little bit of time. The DNA here, if it's unwound, it has 3.4 angstroms, very little, going from one base pair to another. It has the 2 rules, G binds to C, T binds to A, and it turns out that when G binds to C, they're bound via 3 hydrogen bonds. Okay, we'll have to explain, or have to understand what the difference is between the G and C forming the 3 hydrogen bonds and the backbone, it turns out, is covalent bonds. Does anybody know how much energy is in a covalent bond versus a hydrogen bond? Yes? 50 to 100 kilocalories per mole is what? Per what? Per covalent bond. Okay, 50 to 100, you said kcals per mole. Wow. Does anybody really get a sense of that? Is that like... Josh raised his hand. Anybody else get a sense of that?
In comparison, hydrogen bonds have from, I think it was 1 to 7 or something.
Ah,
The color is from.
okay, good. So you can get a relative size that the hydrogen bonds are much, much weaker than the covalent bonds. And that's good. So in general, these are bound via covalent bonds, and they don't break. They're really stable. And if they do break, you know, like over here, it's big problems. It's like, what do you call the doctor? Because when the DNA strand actually breaks, the cell is probably going to die. Where these hydrogen bonds, they break all the time. In fact, we're going to calculate, I think we'll have time, how much do they break? By the way, he mentioned kcal per mole. So per mole. What is a per mole? What is a mole? Yeah.
6 starting instead of the 23rd molecules.
Yeah.
Some sort of atom.
Yeah. Okay. So you have kcals, which I don't get that. Doesn't warm my heart. And per mole, 6 times 10 to the 23rd, that's a mole. That doesn't exactly warm my heart. So we're going to do it in more natural form, where we're going to talk about the energies of these bonds compared to what? Compared to the temperature. KT. K is just what is called the Boltzmann factor. And in fact, I talked to you in the so-called box A as to why we use KT. Okay, we'll come back to that in a minute. Okay, so you have every 3.4 angstroms, you have a new hydrogen bond. So we said in total there's about 3 billion of them, right? So what is the length of the DNA? So we have 3.4 times 10, how many meters? Minus 10? This is the length per base pairs times the number of base pairs we said is 3 times 10 to the 9th. And that's equal to what? That 3.4 times 3 is about 10 times 1/10. So you have in each and every one of your cells, you have about a meter of DNA. A meter. And how big is a cell? Tiny. It's, roughly 5 microns or something like that. So in, let's say, a micron, you have to somehow compact this meter-long thing into like a micron. In fact, we're going to study how do you do that. Yes?
Just curious, what's the cross-section of DNA?
The cross-section, this dimension is about 2 nanometers. It's very small and really long. Though the good question is also, what is the flexibility of the DNA? And in fact, it's called the persistence length. we're going to talk about that somewhat. Okay, so this is a bit more about DNA. That it has what's called a major groove and a minor groove, and you'll get this. Yes?
Is there something like inherently advantageous to the DNA doing this spiral thing, or is it just something it does and we recognize a lot?
That's a good question. Okay. No, seriously, that it just does it. That's the answer that I have. It's kind of a crappy answer, but we won't tell anybody. Yes?
Wouldn't one of the advantages be that it makes it easier for it to compact itself?
Makes it easier to compact itself? Yeah, sure. I agree with that, I guess. Okay, the minimal knowledge, A binds to T via 2 hydrogen bonds, C, G, and binds to G with 3 hydrogen bonds. And what we're going to find is that there are many weak bonds, although it will give a very strong overall structure. So let's actually come to how strong are these? We talked in terms of kcals per mole, but let's do it in terms of kT. And then, because I want to get to the Boltzmann factor. Okay. There are 4 types of chemical bonds. This is essentially sort of the PChem, physical chemistry of biology here. What are the 4 types? Covalent? Okay. Covalent is the strongest. What is second? Ionic? Good. Hydrogen? And what we generally call van der Waals forces. Okay. In general, a covalent bond is really strong. That is, it's about 100 times kT, where kT is the amount of a typical atom in your cell or anywhere else. kT as units of energy. And basically, the covalent gets it from sharing the electrons. Interestingly enough, is light enough to break a covalent bond? He says no. Well, you take a 1 micron long wavelength. So that's in the IR. What wavelengths can you see? What's that? 2 to 700. No, you can't really see 2, but more like 400 nanometers or 0.4 microns to about 700 nanometers, 4 to 700 or 0.4, 0.7. So, okay, take one micron, and that it turns out is 1 eV, electron volts. kT, it turns out, is a 20th of an eV. So from that, you get 1 micron is about 20 kT. And so, in fact, there, 100 kT is equal to 1/5 of a micron, which is about 200 nanometers. So that's UV. You typically see, from the sun, you typically see 300 nanometers, 400 nanometers. So there is enough damage that you can, about break a covalent bond by sitting in the sun. In fact, that's right. They say that you shouldn't sit in the sun. Or, you rub that the sunscreen stuff to basically absorb that, so your body isn't absorbing, so you don't have any covalent breakages. And because we said if you have a covalent breakage that is like of the DNA, your cell is in trouble. Okay, the ionic bonds, they vary tremendously, anywhere from as strong as 100 kT down to only a few kT. Ionic bonds is when you have two different elements like sodium and chloride. Chloride has a lot of electronegativity. It likes the electrons, so it tends to grab them. And the sodium doesn't care so much for the electrons, so that means the sodium has a plus and the chlorine has a minus. And in general, if you have table salt, is it stable? Sure. However, you take some table salt and you add a little bit of water, and it dissolves. Why does it dissolve? Because the H2O is itself highly polar. The H2O, the O likes the electrons, so it tends to be fairly negatively charged. The hydrogen doesn't really like it so much, so that tends to be positive charge. So the negative charge interacts with the sodium and tends to surround it. And similarly with the chlorine, it's negatively charged and has a positive, interacts with the positive hydrogen atoms. So because of this, if a sodium chloride in solution, it will only be like a few kT. So in solution, the ionic bonds tend to be close to kT. And if you're close to kT, you have it. Hydrogen bonds, which are just like what I was talking about, when a hydrogen bond is bound to an oxygen or another atom that really likes its negative, the electrons, it tends to be somewhat positive here and somewhat negative here. And because of that, it can compete very well. And van der Waals, they tend to be the weakest. They are on the order of kT. But in fact, if you have multiple ones, you can actually end up getting fairly strong. In fact, we're going to take a minute and we're going to understand how many people know what a gecko is? So people who apparently don't watch TV, because on TV they've made a real famous gecko. Who's the famous gecko? Geico. You know, they have this like cute little gecko, which is like a little lizard, and he walks around making jokes. And it's supposed to convince you to throw money his way or something. Anyways, what you have in van der Waals, but we're going to discuss the gecko in a minute. You have a simple atom which has its minus and its plus, and the minus in general goes around, though at any one point you'll have minus will be over here and plus will be over here, so that creates a little dipole moment. And if you have another one that just happens to undergo similar, has a dipole moment, and they're close enough that this plus charge will be attracted to this minus charge, and this minus charge will be attracted to plus. And so in general, they'll be a little bit of positive attraction. And so we're not going to get to this. Okay, well, let's take 2 minutes and think about neon and think about xenon. Neon and xenon are, which class of molecules are they in? Noble gases. So they actually don't react hardly at all. They don't react via covalent, via ionic, or via hydrogen bonds. But actually, they can still react via van der Waals bonds. So the question is, what is the difference between neon and xenon? And in particular, you freeze them, and one will have a higher melting point than another. Why is that? And what does it say about the van der Waals interaction between the two? So what I'm going to have you do is look at the person next to you. Say hi. Okay, good. Okay, good. You can introduce yourself. Tell us what's your name. Okay, good. There we go. I like this talking. Okay, so tell me. Okay, stop talking for a minute. What you're going to do is work with a friend who you now, yeah, nice to meet you, and this and that. Have you, like, shaken hands? You know, oh, I was just in France, you know. Okay, tell me about the van der Waals interaction. You freeze them. Which has a higher melting temperature? You're going to have to answer this basically in the next 2 minutes. Go. You freeze neon and you freeze xenon. And they're going to have different van der Waals interaction because of their size. Which is going to be greater or less? So xenon is generally, or neon is in general somewhat smaller than xenon. And so which is going to have less or more van der Waals interaction, and therefore which is going to have more or less interaction to determine their melting temperature. Okay, shut up. Go ahead. Go ahead. Are you talking about this?
We were talking about this before. We were like, yeah, we were sick of it. Yeah, we did it first, now we're and now we're
Great. Go on, tell about your semester. Do you think you know? Yeah. You don't get it.
math in the undergraduate study, like without really intentionally doing it.
Really, that's amazing. Okay. Okay. So, who thinks they know in terms of which one has the higher melting temperature? I just want to get hands. Come on. Come on. A little more. A little more. There we go. Wow, man, you guys kick butt. Okay. What is the answer? Neon, it turns out, it melts or freezes, depending on which way you're going, at minus 246, and xenon at minus 108. So you think you got this?
Yeah. Wait. So which one has the higher melting temperature? You're saying neon? No.
Xenon is at minus 108, which is a lot higher than minus 246. Okay. So you've all determined, because it's the different van der Waal interaction and because of the size of them. Okay, the thing I didn't cover today is the Boltzmann constant, and wait, I know this is bad, but geckos. Geckos are fairly amazing in that their palms over here have lots of really small, like hair-like filaments which allow them to stick such that they can even stick upside down. And in fact, the typical geckos can support a weight of 293 pounds. Okay, sorry about this. This happens, in fact, all the time. And so what you have to do, get out a piece of paper, and you have a minute or two, and you need to evaluate. What was the most interesting thing you learned in class today? What are you confused about relating to today's subject? What would you like to know more about?